So I figured out a good stopping point for this. We won't get too far into this. We'll finish this up on Tuesday. Unit 11, there's two major topics here. One of them we'll talk about lipids, fats and lipids, and the other one, the other half of this is proteins. And the proteins one will really be a good lead-in for Unit 12 when we start talking about nucleic acids. So lipids are these biochemical compounds that contain carbon, hydrogen, and oxygen. Right? But lipids are considered to be insoluble in water. With sugars, we can put sugar in water and dissolve the sugar, right? And we can dissolve the sugar because of all the hydroxide groups that we see. But with lipids, lipids have this long tail. So this is an example of one type of lipid. So this is an example of one type of lipid, and why it's not soluble in water, right? Is because we've already learned, we did an experiment with alcohols. Right? The small chain alcohols we were able to dissolve, right? But the long chain alcohols didn't dissolve. And it's a similar case with these lipids, with these fatty acids, is that here, water's attracted to this, water's attracted to this, but water just is not attracted to this long tail here. Right? So these are what we consider to be hydrophobic. Right? This tail here is hydrophobic. While this here is what we call hydrophilic. All right? So hydrophilic, but energy, fats are really good for storing energy. All right? So we classify lipids into two major categories, right? Fatty acid containing lipids, and then non-fatty acid containing lipids. Right? Those are going to be our steroids. I don't think we're going to get to steroids today. We'll probably stop at waxes some point around there. But we already talked about fats and oils in these fatty acid lipids. So the term fatty acid. Fatty acid lipid. And we notice that fatty acids, it's a carboxylic acid functional group. Right? So our fatty acids here, right, we're going to see some commonality there where they contain this carboxylic acid group. Right? So lipids are just long chain carboxylic acids that we call fatty acids, and they're always going to be in some even number, right, ranging from anywhere to 10 to 12. And we're going to talk about two types in particular, saturated and unsaturated fatty acids. So when we look at saturated fatty acids, I have a photo in the next one that I'm going to get to. I'm going to be switching back and forth here. But here, this is what we consider to be a fatty acid, right? A saturated fatty acid. And what is saturated with? It's saturated with hydrogens, right? Is that here, this has the maximum number of hydrogens this particular fatty acid can have. Right? There's nowhere else to put another hydrogen here. We break that rule of having 4 bonds to each carbon, but here there is nowhere, so it's just saturated with hydrogen. And here, this is what we consider unsaturated, because we can still add hydrogen here. Like if I break this bond here, I can add 2 more hydrogens. If I break this bond here, I can add 2 more hydrogens. So this is what we consider to be unsaturated. Now, as far as unsaturated goes, we have mono and polyunsaturated. There's no diunsaturated, there's no triunsaturated, there's no oligo-unsaturated, right? We have oligosaccharides, disaccharides, but with fatty acids, if they have one double bond, monosaccharide, anything more than that is just a polysaccharide. Right? Whether it has 1, 2, 10, we're just going to, I'm sorry, whether it has 2, 4, 10, right? We're just going to consider those to be polyunsaturated, but one, we will just consider that to be monounsaturated. So as we look at these, right? Saturated fatty acid, and because this has 2, this is a polyunsaturated fatty acid. Now our saturated fatty acids, right? When we form these bonds here, these double bonds, is that we know 2 types, right? We've gone over cis and trans isomers, and cis and trans isomers can either have some trans form here, some cis form. And when we look at unsaturated trans fatty acids, so the reason I've drawn it like this is for a specific reason. When you look at fats, sometimes the fats are solid, like lard. Lard is a solid, right? Lard happens to be a saturated fatty acid. But even some unsaturated fatty acids, like I can put this double bond here, let's put this one here, this one here. I'm going to put 3 in this one over here. And even with a double bond like this, right, this can still pack in very nicely. Right? And this is the case with fats that we see are solid. Right? Now, fats that are in liquid form, right? When we look at cis fatty acids versus trans fats, trans fatty acids, trans fats, if we could look at some cis fatty acids, and that double bond, when we look at that cis form, it creates kind of a kink. So when we try and bond cis fatty acids, or try and stack them anyway, is that these are just not going to stack together there's no way that these are going to stack together very well, right? Because they're just misformed here because of this cis bond that we see. Right? But this kind of helps explain why we see some fats as liquid, right, and some are solid form. Now these are just common saturated, unsaturated. You don't have to memorize this list by any means, right? But we do need to know some things about it. This number here in particular, and I'm going to add some information that I feel is missing here, okay? But here, the 12 represents the 12 carbons, and the 0 represents 0 bonds. That's why it says 12, and then we see this colon 0. Just like this has 2 bonds, so 18 carbons. This is 2 carbons here. And then let's take a look at one of these fatty acids here. I'm going to pick one that has a double bond. And let's take a look at lard. Nope, we need a double bond, oleic acid. CH3, we have 7. 7 more. And then we have CO2. I'm going to rewrite this in just a second. Remember, this here represents this. Represents carbon, like carbonyl, oxygen, and hydrogen. This is the same thing as writing this, COOH. COOH, carboxylic acid, carboxylic acid, carboxylic acid, right? That all represents the same. So I'm just going to redraw this real quick because this is what I want to see instead. There we go. So there's my COOH. Now we're going to count the carbons here. So this is carbon number 1. And this is carbon number, what are the carbons here that are in these 7? Carbon numbers from what to what? From carbon number 2 to what? To 8. So this is carbons 2 through 8, which means this is carbon number 9, this is carbon number 10, and this would be carbons numbers 11 through what? 17, and then this is carbon number 18 here, okay? So here it says that it's 18, 1. But I feel there's some information missing from this because if I give you this, you can draw the fatty acid, but do you know where to put the double bond, right? So what we do here is this represents, I know it represents heat, but remember it also represents change, right? Like change in temperature, we use that triangle, right? So it represents change, so we're gonna put 9, which means that 18 carbons long, there's 1 double bond, and that double bond occurs at carbon number 9. Right. So I think that's just a small piece of information that might be missing, but that might come across on the exam. Hint, hint, right? Might come across on the exam, and we might see that. Okay. Now, the other thing that we do need to learn how to do, we're going to be doing some reactions. I don't know if we're going to get to those reactions today or if we're going to get to them on Tuesday, but we have to be able to rewrite this where this end is the carboxylic acid functional group. So I'm just going to spin this around here. Okay? I'm going to look at this, and I'm just going to write this backwards. So my CO2H is my carboxylic acid, which is what I have here. And then what comes next are 7 CH2s. CH2, and there's 7 of them, and I have the same thing here. And then I have CH and CH, so I'm going to write CH, double bond, CH. I have 7 more CH2s. And then lastly I have CH3. So all I've done is I've done my little magic marker trick and I've just turned it around. But when we're doing reactions, we need to be able to do this even though the table over here has it written the other way. So let's look at the linoleic acid. So I'm going to write linoleic acid the same way that I've written it up here, but I'm going to just draw it the other way. It's important we learn how to do this. So we're doing linoleic acid, the safflower there. I have my CO2, so here's my carboxylic acid. Then I have 6 CH2s. And then I have, let's see, we're going to go with CH2. And then we have CH, double bond, CH. I'm going to close that parenthesis. And there's 2 of those. And then we have CH2, there's 4 of them. CH2, we have 4 of these, and then there is CH3. So all I've done is I've just taken this one here, but I did turn that around and write that the other way, because again, we are going to be doing reactions using these, and we just need to know how to write it this way in order to properly get the answers that we need. Now, here, I need to know, first off, how many double bonds there are in the structure. There's 2. Because I have 2 of these, it means there's 2 of them. And now we need to decide where those are. So let's count these carbons. Carbon number 1, which means this is carbons numbers 2 through what? There's 6 of them, so 2 through 7, right? 2, 3, 4, 5, 6, 7, right? 2, 3, 4, 5, 6, 7. 2, 3, 4, 5, 6, 7. 6 of them. 1, 2, 3, 4, 5, 6. CH2, 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 CH2. Okay? So this is 2 through 7, which means this is going to be 8, 9, 10. But I have 2 of them. 11, 12, 13. Does that make sense? And then here, this is going to be 14 through 17, and then this is 18. So as far as writing this down over here, well, the change occurs on what carbons? Carbons numbers 9 and 12. So if you're given this and you had to write your own, anybody should be able to draw this fatty acid, right? If you're given this information here, you have no idea where to put those double bonds. But if I have this information, here's my carboxylic acid, carbon number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18 carbons. And my double bond begins on 9 and 12. Double bond begins here, double bond begins here. So this is the structure of what we see here. All right. All right. So animal fats, vegetable oils are what we call lipid esters. Right. Lipid esters, we call them triglycerides. You might hear the word triacyl glyceride. But essentially they are just esters. And let's try and remember an ester. Right? This is the functional group of an ester. We have this carbon, then an oxygen, and some carbon chain over here, some carbon chain over here. But it's this carbonyl oxygen that we see. And that's what we see in an ester. So an ester essentially is 3 carboxylic fatty acids that are bonded together using glycerol. And if I was taking an exam, I'd want to know what glycerol looked like. Right? This is glycerol. I'm going to write glycerol down a couple different ways. So glycerol can look like this. Glycerol can look like this. Right? Glycerol, this is also glycerol. Expanded structural formula, this is also glycerol, right? But glycerol, 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 and then what we do is we just attach a fatty acid. So let's just kind of review an esterification reaction. So this is esterification. Is we take some carboxylic acid, plus we take some alcohol. Let's do it. Carboxylic acid plus some alcohol. Let's do this. Let's take an alcohol plus our carboxylic acid. And just so we know it's the same reaction, I'm going to do it this way as well. It doesn't really matter left or right, but I think it's more understood if I write it this way. We added some acid to that, we're going to add some heat to that, and we're going to get our ester. It's the same product, all I did is just spin it around. But this is an esterification reaction, right? We take some carboxylic acid, we add an alcohol, carboxylic acid, we add alcohol, and we get this ester, right? That's our ester. And that's all that a triglyceride. Triglyceride just happens to be 3 esters on the same molecule, right? Is that this is an ester, this is an ester, this is an ester. Carbonyl, right? Oxygen, and then carbon. So it is our ester here. So essentially, right, they are just lipid esters, but we call them triglycerides. And we just take glycerol. We take glycerol here. Here's our glycerol backbone. Here's our glycerol backbone. Is that we form that ester, is that this is a fatty acid, fatty acid, fatty acid. If that glycerol being the backbone of it, right? So how would we make this triglyceride? Well, we would take an alcohol. We would take some alcohol, OL. Glycerol ends in OL. There just happens to be 3 alcohol groups on there. But that's how we make these triglycerides, is we take glycerol, this is our glycerol backbone, alcohol, alcohol, alcohol group, glycerol, and then we take some fatty acid, carboxylic acid. So carboxylic acid plus an alcohol, and we form this ester. And it's the same thing we did here, carboxylic acid plus this alcohol, and we form our ester, right? Carboxylic acid plus this alcohol, we form this ester here. So this is an esterification reaction. And we get 3 water molecules here. Now, the fatty acids don't necessarily need to be the same. In this case, they are. But they don't need to be the same fatty acid, as we could have different fatty acids. This happens to be a saturated fatty acid. This one's mono unsaturated, and this is a polyunsaturated fatty acid. We know that because it has 2 bonds. But we can see here that it doesn't necessarily need to be the same fatty acid on that triglyceride structure. So how we make soap. Let's see what else we have here. Yeah, I think let's do saponification, and then we'll probably cut it off from there, right? So saponification or soap making. Right? Unfortunately, we don't have time. I think that was lab 12 or 13. We do end up making some lotion or some soap, which is simply just a triglyceride. Saponification. A fat or oil plus a strong base. Not a strong acid, not a weak acid, not a weak base, but a strong base. It's bolded, it's in red. Why would I do such a thing, right? In order to undergo saponification, we need to have a strong base, right? So the sodium salts, right, those fatty acids, those are what we call salts, but let's take a look what we get from that. So we're going to get glycerol plus we're going to get some soap. So here is some triglyceride here. All right. This happens to have the same fatty acid, but this is our glycerol backbone. Right, here's our ester bond, but this makes up our triglyceride. Now, this is sodium hydroxide, strong base. But we do need 3 moles of this. This is a similar reaction where we did something we called base hydrolysis in organic chemistry. So if you recall base hydrolysis, so we're going to take some ester. We're going to add a strong base to this. And we break that apart. We get alcohol, plus we get this carboxylate, right? It's the same process, but it just happens 3 times, right? For this one, I only need 1 mole of sodium hydroxide because there's only 1 ester bond to break. But here, there's 3 ester bonds. I have to break this ester bond, and I have to break this ester bond, and I have to break up this ester bond. So we're going to need 3 moles of sodium hydroxide in order to do that. And what we do here is we get glycerol. When we break this bond here, we get alcohol. We break that bond here, and here's my alcohol, which is glycerol. And we're going to get this carboxylate. Notice how it's sodium palmitate because it's a carboxylate, right? Here's sodium, here's palmitic acid, acetic acid, acetate, palmitic acid, palmate, right? So here we have sodium palmate, which is a soap, but we do have 3 of these, right? So there are 3 of them. That's why we put the 3 in front of it, that coefficient. So we do have 3 of those there. Now let's do one of those reactions. So it says a particular glyceride contains myristic acid, palmitic acid, oleic acid. How many moles of sodium hydroxide are needed to saponify 3 moles of this hydroxide? Write the complete balanced equation. This might be one of your homework problems. I'm not quite sure. It might be one of the homework problems. So just kind of make a little note. When you get to that homework problem, take a look at this here. Right? But here's myristic acid. Right? There's palmitic acid, and this is oleic acid. And let me explain how we got to that point there. When we're making these triglycerides, everything happens from this glycerol backbone. So here's our glycerol backbone. CH2, CH, CH2. Here is glycerol. Now with glycerol, the first one, let's go back to this table. Myristic acid. So this is the reason why I had to spin it around. This is myristic acid here. I need CO2H. This is, I'm going to take a little shortcut here. This is my CO2H. But I need to put this together with glycerol to make the triglyceride. So what I'm going to do is I'm just going to remove this OH here, and I'm going to remove this hydrogen here, and then I'm just going to take this and start it here. So I'm just going to go like this. So I have my ester. Now I'm just going to continue. I got 14 CH2s. CH2, there's 14 of them. And then I have a CH3. The next one was, I believe was palmitic acid was the next one. So again, I'm going to repeat that same process. I'm going to erase this. I'm going to put in my carbonyl. That represents the CO2OH, right? 14 CH2s. Oh, myristic acid. Oh, this one had 12, not 14. Let's fix that. This has 12. This one has 14. CH2, there's 14, and then there's CH3. And the last one, I believe, was oleic acid. Palmitic and oleic acid. So we're going to take oleic acid, we're going to do the same thing. Carbonyl, oleic acid, I have CH2, there's 7 of them. CH2, I have 7 of them. And then this is where we have CH2, that double bond right there. And then we have CH2 again, and there's 7 of those. CH3. And this should match what we see here. So that's all we've done. This is the same thing that we just did, right, on the board as we make this triglyceride. So using that table, you can make any triglyceride just following the same process. Now we need to saponify this. In order to do that, we need a strong base. So we're going to use sodium hydroxide. Because I have to break apart 3 ester bonds, we're going to need 3 moles of sodium hydroxide. We take glycerol, so we get glycerol back, right? We break apart our ester, we get our carboxylate, and we get an alcohol. So we happen to get this alcohol back after we break that apart. And the rest of it, so here, if I remove glycerol from here, I've just removed glycerol. And now we're going to add the sodium hydroxide. We're going to get this O here, negative, 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 Na plus, Na plus, Na plus. These are our carboxylates. Carboxylate, carboxylate, carboxylate. So here, we get that carboxylate up there. We just separated that. We get this, and we get this. So this is the complete balanced reaction. We take these 3 ester bonds. We do saponification on it, which is basically just base hydrolysis. We get our alcohol, plus we get these carboxylates, which is our soap. All right. We can cover this real quick. All right. I think we'll stop it when we get to waxes here. So we know what hydrogenation is. Remember hydrogenation. This is an addition reaction. We have this double bond here. We're going to add hydrogen gas across that double bond. Let's use palladium there. So we break the double bond. We break this double bond, and then we're going to add this hydrogen across it. Right? So that's what hydrogenation is. Every time I break a double bond, I need 1 mole of hydrogen gas. Every time I break 1 double bond. So here, because I have 2 double bonds, how many moles of hydrogen am I going to need? 2, because I have 2 double bonds to break. If I had 3 double bonds, I would put 3 moles of it. So looking at this reaction here, and it's the same here. We're not going to, this is already built. This is oleic acid, linoleic acid, palmitic acid, but this is our triglyceride. You can review the lecture video or just, remember how we build it. This is our glycerol backbone, and then we take a look at the table and we add each of those fatty acids to it. So this is our triglyceride here. How many moles of hydrogen am I going to need in order to break all these double bonds for hydrogenation?
Would be 6.
So let's count. We're not looking at these double bonds. We're just looking at the double bonds in the tail. So How many? 3. I have 2 here, and I have 1 here, 3, and these double bonds we won't worry about. Right, we just the ones that are in that hydrophobic tail that we have there. So we would just need 3 of those, so 3 of those, and then we just simply break it up. And you'll notice that this doesn't have any more double bonds, is that they're just a bunch of CH2s, right? So we don't need to, we could do it this way. We could, if I'm gonna hydrogenate this, I could put CH2, CH2, or I could just put 7, 14, 15, 16 in one, and then, so this is just a little shortcut that we took here. But we break that double bond here. We have 6 plus 4. We add these in here, right? We're gonna get 16 of them. But that is our hydrogenation, and that brings us to waxes. So we'll continue with waxes on Tuesday, and then we'll finish off just units 11 and 12 on Tuesday. So let's break until 11:10. I'll get the supplies ready. It's not a very lengthy lab, but I think we're good for today.
All right. Top one. Don't make me use this big ugly one over here. Good. All right. So we ended up, we finished this section here. We're now into our next fatty acids, waxes. We're not going to talk about any reactions with waxes. Waxes aren't very exciting. They are still considered to be fatty acids. Except these are also, they contain ester bonds. Right. So here, I'm just going to draw a small ester. Right. So remember this ester bond that we have here from organic chemistry, right? And then we would call this propyl acetate, right? So this would be considered an ester. So waxes are just long chain esters, right? This is a very small ester. Again, this is isopropyl acetate. And then we would have what's considered to be a wax. So wax is just a very long ester. Right. Containing 1232 carbons. Instead of glycerol there. But this here is just what we consider to be a wax, you know. And we can see that we can just kind of stack these on top very well. We're not going to put any branches on these in particular ones here. No branching, no cis, you know, trans bonds that we have to worry about. Just be able to identify that this is a wax, right? That's kind of all we need to really consider when we're looking at waxes. That they're long. Then we take this long chain alcohol here. We create some ester bond through esterification. But we just need to be able to identify, if we see this, well, is this a fatty acid? Yes, but what kind of fatty acid? Because we do have different types of fatty acids, right? We have fatty acids that look like this. Actually, I'm going to take a little shortcut here. And we'll just kind of keep this on. What else do we have? That's another one that we've learned. One more that we've learned. Right. Be able to identify different types of fatty acids, right? So in a lineup, could we pick one of these out to be our wax? Absolutely. This is our wax. How do I know it's a wax? Because it contains an ester bond, and then I have two very long carbon chains, right? What's the name of this fatty acid? Right, we have to be able to identify these. Brian, what do you think? This is a wax. What is this one? What is this one, Bryant? This is glycerol backbone, but the name of this particular fatty acid is a triglyceride, right? So we have to be able to identify the types of fatty acids, right? Can somebody name this fatty acid up here? Does have a way we can identify it. What would we call this? Zuri. A saturated fatty acid, absolutely. What would we call this down here? Melissa. Monounsaturated fatty acid. And then if we had two bonds on there, we'd call it a polyunsaturated fatty acids. But other than just a mono fatty acid, how else could we also identify this particular one versus this particular one? Montalene, what do you think? This is a polyunsaturated fatty acid, but what else could we use to identify this particular fatty acid? The double bond, and which one would it be?
One closest to the.
So, which one would it be?
Oh, okay. Trans.
So, trans, let's take a look at it. See how nice and straight that is? And see how this one is, oh, not that straight.
Oh, because it's
So cis. So this would be a poly cis unsaturated fatty acid versus a trans mono unsaturated fatty acid, right? So by looking at them, we do need to identify like the family of fatty acids that they do belong to. And let's move past these waxes. So I didn't really have much to say about these waxes. The next type of fatty acid is what we call phospholipids. So phospholipids are similar to triglycerides. Right? I'm going to leave this one up here. So this is a triglyceride. And we'll notice how these phospholipids are similar, right? We still have our glycerol backbone, right? We have this fatty acid here, which would be a saturated fatty acid attached to it. And then we have this monosaturated fatty acid attached to it. But here, we don't have a third fatty acid. Is that third fatty acid is replaced with this phosphate here, right? We have this phosphate group here, and then we have this. So we don't really have our ester bond that we're normally seeing. So we end up with these structures here, and this end. Let me go back to this other slide here. Right? We've talked about these ends here, right? These tails right here are what we consider to be hydrophobic. Wants nothing to do with water. Water and those long tails just don't mix. But this phosphate, on the other hand, this phosphate with these two oxygens here, right? This nitrogen that has this positive charge here, right, is that this part is hydrophilic, right? Hydrophobic, doesn't like water. Hydrophilic, very attracted to water, right? So when we put phospholipids into an area with water, right? If we put them into an area with water, we have this phosphate portion, and then we have those two tails, right? And when we put them in water, these phospholipids will orient themselves autonomously, just all by themselves, not much work, but what they create is this barrier. They create this barrier here. If you've taken some biology, if you've ever heard the term phospholipid bilayer, well, these are phospholipids, right? This is a phospholipid here. And it forms this barrier that we call a phospholipid bilayer, right? This is the idea, this is the thought process behind what we consider to be cell membranes, right? Is that here we have water, but they form this barrier here where no water can exist here, right? So here it is, our cell membrane, and they just kind of orient themselves here to form that cell wall, right? Or I'm sorry, not that cell wall, but that cell membrane. All right. So we've covered all of our fatty acid lipids, right? We have saturated fatty acids, unsaturated fatty acids. We have cis-trans unsaturated fatty acids, polyunsaturated fatty acids. We have our waxes, triglycerides, and our phospholipids, all under the category of fatty acid lipids. So now let's look at the only non-fatty acid lipid that we're going to talk about in this class. There are others, but this is the only one we're going to talk about in the class. And these are what we consider to be steroids. So steroids are considered non-fatty acid lipids. Right? But the reason that we do consider them lipids is because of the amount of carbons that are there. Right? Is we know that lipids, fatty acids all contain a lot of carbons, and so do steroids, but they don't fall under the category of fatty acid because there's no carboxylic acid and there's no ester bonds here. Is that here we just have what we call the steroid ring system. The steroid ring system that contains 16, 17, right? 17 carbons in this steroid ring. But the steroid ring system always looks like this. Always, you know, cyclohexane, 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 cyclopentane, and there's just a lot of carbons in here. So all of these carbons are all hydrophobic, right? Water doesn't want to be near those. But what makes these steroids different are the branches that we can put onto this steroid ring. Right? This is cholesterol. Cholesterol is a steroid. Right? But it's just these different branches that we can put on here, right? We have this methyl group here, we have this double bond here, we have this alcohol group here, there's another methyl, here's this big alkane branch here. And then we see differences with other steroids, right? Androsterone, progesterone, estradiol, testosterone, estrone. But we can see here the difference here, because we still have methyl, this has methyl. Here's our ketone functional group. We have a benzene ring over here, so we have phenol over here versus cyclohexanol, right? We have this methyl group here, but we don't have that methyl group here. Okay. So there's just little differences, but these differences make all, you know, they make all the difference when we're looking at different types of steroids, right? Here we have phenol, here we have phenol, we still have the methyl group, we still have the methyl group, but instead of a ketone functional group, here we have an alcohol functional group. So this is our difference between estrone and estradiol. Now, I won't ask you to memorize all of these structures, right? You don't have to memorize these structures, but you do have to know the names of them, right? You do have to know that cholesterol, testosterone, progesterone, estradiol, you do have to know the names of them and know them by name that in fact they would be steroids, right? If I gave you a list of just different substances, could you pick out which one is going to be the steroid, right? So you don't have to know the structure of them, but you definitely have to know the names of these steroids here. Now we're getting back or into proteins. This will be a good lead-in for Unit 12. Unit 12 is pretty short, but I think talking about proteins today and then covering Unit 12, we shouldn't be able to make a really good connection between those two units. Right? So what are proteins? Proteins are large, complex molecules composed of amino acids. Right? They all contain carbon, they all contain hydrogen, they all contain nitrogen, and there's 20 different amino acids that make up all of our proteins. Right? All the proteins that we make, biologically are composed of 20 different amino acids. And the shape of the protein is really important, right? So all proteins don't have the same shape because they use different amino acids, and these amino acids have different shapes. But whatever the protein is, there is some commonality between all of these different amino acids. All right. So here, every single amino acid right? Because proteins are, this is an amino acid, and proteins are consisted of chains of these amino acids that are bonded together. So every single amino acid is going to have this carboxylic acid group. Every amino acid is going to have this amino group, right? That's just this nitrogen here. All of them are going to have what we call an alpha carbon. Okay? So this carbonyl that's right here, the carbon that's next to the carbonyl is considered to be the alpha carbon. So if this carbon is the alpha carbon, then this nitrogen would be the, let's try this again. This is the alpha carbon. This would be the, what was that? It is the amino group, but let's kind of listen to what I'm saying here. This is the alpha carbon. Adam, this is the, well, this is alpha. It's carbon. This is because it's the first carbon next to this carbonyl, right? So here, we're not talking about alpha beta as far as isomers are concerned. We just need the name of this. This is our alpha carbon, and this would then be our alpha nitrogen. Perfect, right? So this is our alpha nitrogen. So alpha carbon, alpha nitrogen, and then off of the alpha carbon, we have what's considered the side chain. And the side chain is what makes all of these amino acids different, right? It's always that side chain. We can see different side chains here for leucine, isoleucine, methionine. And we can see, right, here's our carbonyl, this is our alpha carbon. Montylene, this is our alpha nitrogen, yeah, so this is alpha nitrogen. And this is our side group, right? This is our side group. So we see the difference between isoleucine and leucine is that here, this methyl branch is on a different carbon, right? And that's the difference between leucine and isoleucine, right? Methionine here, well, we have sulfur involved, right? So these R groups, these side chains, are what make all of these amino acids different, right? They function differently, they have different functional groups, they build different proteins. And we'll talk about, they're usually categorized. We categorize them non-polar, polar acidic, polar basic, polar neutral. So here's all of our non-polar amino acids. And they're non-polar because in the side chain, we're not looking at the carboxylic acid group, we're not looking at the amino group, we're looking at the side chain. And these side chains don't contain any oxygen, right? So there's no hydroxide here. But here, right, these are what we're going to be considered to be non-polar amino acids. Because they have all those carbons and they are making non-polar bonds. Then we have what we call our polar acidic and our polar basic. So polar acidic bonds, right? Here's the functional group, right? This is a carboxylic acid, right? And then we have our amines. So carboxylic acids are acidic, and amines, which are derived from ammonia, are basic. So here, the reason, right, the name of it, aspartic acid, glutamic acid, these are polar acidic amino acids because the functional group, the R group right here contains this carboxylic acid functional group, right? And here we can see this is an amine, right? This is an amide, right? This is an amine. Right? So here we consider these to be polar basic amino acids, right? Lysine, arginine, histidine. And then we have our polar neutral amino acids. So polar neutral amino acids, we see this oxygen hydrogen or this sulfur hydrogen bond going on here, and we would consider these to be polar neutral. is that here we're not. There is some argument here. We are going to consider these to be polar neutral versus polar basic. So there is kind of some argument there regarding those there, but we're not going to really worry about those. If I wouldn't put something questionable on the exam, okay? So we can also consider that. Now, two of these are in fact even more unique. Like we already have the R groups that make them unique, but there's two other amino acids here that are very important. One is glycine. Glycine, you know what, I might have a slide of it. Let me see if I have a slide of this one. Oh, perfect, I do. Glycine is the only amino acid where the alpha carbon is not chiral, right? It's not chiral because it contains this hydrogen, it's bonded to this hydrogen, this nitrogen, and this carboxylic acid over here. So we only have 3 different groups, right? And let me draw it out in expanded structural form so we can make sure that we see that here. So here's carbon, it's bonded to this hydrogen, it's bonded to this hydrogen, and then it's bonded to this amine group here, and then it's bonded to, right? So we have this, we have this, we have this, we have this, but because these two are the same, this is not a chiral carbon. This is in fact achiral, right? Here, if we're looking at alanine, well, all of the rest of these amino acids, that alpha carbon is chiral. We have this, we have this, we have this, we have this, makes this chiral, right? So glycine is the only amino acid where the alpha carbon is not chiral. Now, the other one we have that's pretty unique is proline. Proline is the only amino acid where the R group, that side chain, wraps around and attaches itself to the alpha amino group. And I'll show you that here. So here's the alpha carbon, right? Here's my carbonyl, here's my alpha carbon, and then off of here we have our alpha nitrogen. And this R group wraps itself around and bonds itself to this alpha nitrogen group. And it's the only amino acid that will do that. There's no, there are other amino acids, I'm not talking about the only one that has ring structure, but I'm talking about from the alpha carbon attaching itself to the alpha amino there, right? When we look at any other, is that yes, this has a ring structure, but this R group doesn't wrap itself back around and attach itself to that alpha amino group that we see there, right? So that's what makes proline just a little bit more unique than the other ones there. All right. For this one as well, we do have D and L isomers that we've already looked at. And for carbohydrates, right, we're looking at the hydroxide group being on the right or the left. Well, for amino acids, we're going to take a look at the alpha amino here, right? Here's our carbonyl. Here's the alpha amino group. And then this particular nitrogen, the amino group here, being on the right or the left, right? This is on the right side, it's L. This is on the, this is on the left side, it's L. This is on the right side, it's D. And again, we see that with cysteine. So there are different ways to draw these structures. Right? So we might see them drawn a different way, but in order to identify D and L, again, we're not going to look for the hydroxide group, we're going to look for this amino group being on the right or the left when we're looking at these structures here. Now, I mentioned it before, right? Every amino acid has this acidic group here, has this basic group here, and these amino acids, let me just draw, I'm going to draw alanine. Alanine is pretty easy to work with and draw. My alpha carbon. This portion here is acidic. This portion here is basic. So on this amino acid, we have part of it that's basic, part of it's acidic. If we take a look at this reaction here, if we ionize these, if this ammonia is my base and water is going to act as my acid, we know that this is an acid because it's carboxylic acid functional group. This is acetic acid. Acetic acid, water is acting as my base. Do acids donate or do they accept hydrogens? They donate. So this hydrogen gets donated and we end up with, we go from acetic acid to acetate. We've donated that hydrogen and we get acid and its conjugate base. And for ammonia, well, bases accept hydrogens. Base and its conjugate acid. So bases can accept hydrogens and acids donate hydrogens, which means that amino acids, this basic end, will it accept or donate hydrogens? It will accept a hydrogen, right? And here, this is going to donate hydrogens because this is basic, this is acidic. So we know that acids neutralize bases and bases neutralize acids. Right now, this is a neutral amino acid, right? Neutral amino acid, there is no charge on it, right? But there's three things that can happen with an amino acid. With the pH is down to 3, is this acidic or is this basic? So we have neutral is 7, and then anything below 7 is acidic. Anything above 7, basic. And physiological pH, right? We're gonna stick right around 6. right? Not 7, but right around 6. So we'll consider this to be neutral. Okay. Now, here, if we take this amino acid and put it into an acidic solution, acids neutralize bases, bases neutralize acids. So some things can happen here. Now, the acid, is the acid going to react with the acid? No. The base is going to react with the acid solution. So the base and the acid here. So if we put this into an acidic solution, we're going to get, let me put this here. Hydrogen proton donor or acceptor? Acceptor. So just like we went from NH3 to NH4 plus, we're going to go NH2 to NH3 plus. And now we have an acidic amino acid, right? It's acidic because I see this plus sign, which reminds me of NH4, ammonium, and I know ammonium is a conjugate acid of its base, ammonia. Acidic, the carboxylic acid end is acidic, is that this is now an acidic amino acid. And when we put this into a basic solution, well, the base isn't going to react with the base is going to react with the carboxylic acid end. Proton donor, so we're going to get, this end remains unchanged. But we're now going to get this basic amino acid. Because here's the carboxylic acid end, and here's my carboxylate. Right? So we end up with a basic amino acid. And this is the fourth option here, is that we still need something neutral. This is what we call a Zwitter ion. Zwitter, we have this charge here, and we have this charge here, and this charge offsets this charge. So a Zwitter ion, this is an ion, a Zwitter ion contains both positive and negative charges. Right? This one just contains a negative charge. This is a basic amino acid. This one contains a positive charge. This is an acidic amino acid. And this is our Zwitter ion, and it contains both positive, negative charges there. So that's what we're going to consider to be. It's neutral overall, right? Zwitter ion is neutral overall, but it does contain both positive and negative charges there. So that's going to be our Zwitter ion. You might have a homework question related to something like this. So when you get to that question, kind of look at your notes over here, because I think that'll help answer that particular homework problem. All right. Now, we introduced the idea of proteins earlier, right? Proteins are chains of amino acids. We talked about polymers in Unit 10. And polymers, like starch, glycogen, amylopectin, right? Those are all just chains of glucose. So we take one glucose, chain it, another glucose, chain it, and we come up with these polymers. So carbohydrates are these polymer chains of glucose, of carbohydrates. Proteins are chains of amino acids. Okay? So we put all these amino acids together and we come up with a chain of proteins. Now, for small proteins, we can call them dipeptide, tripeptide, pentapeptide, but there comes a point where there's so many that we're just going to call them proteins, right? So this dipeptide is just a small protein. It is a protein, it's very small. We're going to call it a dipeptide, right? If there was 5, pentapeptide. If there was 6, hexapentide. But I think at some point, probably around 10, we're just going to call them proteins, right? We're not going to call them peptide chains anymore. We're just simply going to call them proteins. And again, this is a protein. Even though it says dipeptide, it's still a protein. It's just a very small protein. So in order to make a protein, they undergo dehydration. Dehydration reaction, when you guys dehydrate, you lose water. When we dehydrate these to form this bond, we also lose water, right? As this gets removed, this gets removed. Here's our H2O, this is water, it loses water, and it creates this peptide bond. If we go back to carbohydrates, carbohydrates also have a particular bond, right? The bond between two carbohydrates, our oxygen bridge, this is our glycosidic bond, right? Same thing, dehydration, we bond to glucose units together and we get this glycosidic bond. Dehydration, we bond two amino acids together, we get this peptide bond, okay? So amino acids will have this peptide bond between them, and this can be reversed, right? We undergo dehydration, we make the bond. Hydrolysis, we break the bond, right? We did that in we did hydrolysis with the sucrose the other day in the lab, right? Is that sucrose wasn't a reducing sugar, but we take some acid, add a little heat, we separate those two units, we break that glycosidic bond, and all of a sudden fructose glucose become reducing sugars. So here, hydration is we can add water across this bond and then we can break that peptide bond up. So a concept here that we look at is both of these. This is glycine. Here's our peptide bond. Here's alanine. Right? Glycine. There's no chiral carbon here, and then alanine just has this methyl group coming off of that alpha carbon. But glycine bonded to alanine, and alanine bonded to glycine. These make two different peptides, two different proteins, two different dipeptides. Even though they're both using glycine and alanine, these two are different functions. If we take a look at this here, well, I have this on your right side, there's this carboxylic acid in here, and the alpha carbon has a methyl group on there. And even if I spin this around, if I take this molecule here, this peptide, and I spin it around, next to the alpha carbon, I still have this methyl group here. And if we take a look at this dipeptide, well, that alpha carbon no longer has methyl there. Right? So these are two different peptides. So as peptides get longer, right? As peptides get longer, when we look at peptides, this is how we always just kind of view our peptides, right? Is that on your right-hand side, you're always going to have this carboxylin. We can call it the C terminus as well, okay? So if we say C terminus, that's the same thing as our carboxyl end, it's our carboxylic acid end, and then we have our amino end, or we call this the N terminus, right? So N terminus, C terminus. We're always going to see it that way. N terminus on your left, C terminus on your right. You don't have to memorize all of these amino acids, but you do need to know the 3-letter abbreviation and the name of that amino acid. That you have to know. Some of them are easy. Methionine, MET. Valine, VAL. Alanine, ALA. Isoleucine is not ISO. Isoleucine is ILE. Aspartic acid, ASP, pretty simple. But asparagine, ASP, no, it's not ASP because that's aspartic acid. Asparagine is ASN, right? Glutamic acid, GLU. Glutamine, GLN, right? So you do have to know the names and that 3-letter abbreviation for the exam, right? But you don't have to know what it looks like. You do have to know the name and that 3-letter abbreviation. If anybody wants to memorize all these, in biochem over the summer, I teach 207, and in biochemistry, we memorize all of them. It's a blast, right? We have lots of fun memorizing all 20 of those and having to draw them all out on a test. It's great. Good times, good times. Okay. So, to make things a little simpler, right? Chemists, we're always making things simpler. I got to do it quicker, faster, better, easier, right? Is that instead of drawing out what phenylalanine looks right? Glycine, alanine, valine, serine. Instead of drawing them out, we just use that 3-letter abbreviation, right? And if we've memorized them, well, then we know what the structure looks like. But if we didn't memorize them, we can look it up in a book, right? And then we can find out what it looks like. So, but you at least have to know that 3-letter abbreviation to know that this is phenylalanine, glycine, alanine, valine, serine in this particular peptide. So this peptide contains 1, 2, 3, 4, 5 amino acids. What could we call this particular peptide? If I had 2, I'd call it a dipeptide. But since I have 5, what do we call it, Azalea? Pentapeptide, right? Pentapeptide, right? And the bond between this, right? Kayla, what's the name of this particular bond between the peptides? Peptide bond, right? So here's our peptide bond between them, and then we have these amino acids, and this makes up our pentapeptide, which is still a protein, right? It's still a protein, it's just a very small one. And this particular structure here, this is what we call a primary structure. The primary structure gives you one piece of information. It gives you the order of the amino acids, right? So here we know this particular protein, the order of amino acids is phenylalanine, glycine, alanine, valine, serine in this structure here. We don't know what the structure looks like, but we do know the order of those amino acids, right? And that's important because I showed you glycine and alanine, and then alanine and glycine, and that does make a huge difference. That order makes a huge difference there. So this is what we call our primary structure. As these start to get larger, right? As these start to get larger in size, they start folding and creating different shapes, creating hydrogen bonds with other R groups and the amino acids. And this is what we consider to be a secondary structure, right? This is a secondary structure. This is considered an alpha helix. And what happens when they get so long like this is we start getting interactions between the R groups. The side groups here, oh no, not between the side groups, between the carboxyl and the amine. So I spoke out of turn there. So if we look at an amino acid, because of the way it twists and folds, we create a bond between the alpha amino group and that carboxyl group, and they form this hydrogen bond. And that's how this alpha helix stays in that shape, is that this oxygen here on the on this carbonyl is going to bond not to this amino group, but it's going to bond to another amino group in this chain. So here's our carboxyl group, alpha carbon, alpha nitrogen, and then it's making a hydrogen bond to, let's find it here. This is another amino acid here. There's another amino acid here. There's another amino acid here. And another amino acid here. So 4 amino acids away is this hydrogen bond is occurring between that carbonyl and this amino group here. Is that 4 amino acids away, it's creating a hydrogen bond. And 4 amino acids away, it's creating a hydrogen bond and another hydrogen bond. And these hydrogen bonds are what keep this alpha helix shape. So again, this is known as a secondary structure. Primary structure, we know the order of the amino acids. Secondary structure, we start seeing what we call these alpha helix structures. And we also get, less common, but we also get what's called these beta pleated sheets. Right? The paper looking structure is just more for visualization for these sheets. So there is no paper looking structure holding these together, but this is just more for visualization to see why it's considered to be a beta pleated sheet, right? It's like when you fold a piece of paper over and over, try to make those little fans, it has those little pleats, right? So that's what we see there. Less common, but still. Now, as far as beta pleated sheets, there are 2 types of beta pleated sheets. Right? We consider these to be anti-parallel, or they can run parallel to each other. Now, as far as parallel, let's look at anti-parallel because there are some differences here. Right? When they run anti-parallel, they're running in opposite directions. Right? Here's our carboxyl end, and the blue will usually represent nitrogen here. So this is carbon with its oxygen, and then here's the amine group and the nitrogen. And this is running the opposite way. So carbonyl, alpha carbon, nitrogen. Carbonyl, alpha carbon, nitrogen. Right? So these are running in opposite directions here. Okay? Opposite directions they're running. And the other thing to notice is those hydrogen bonds are pretty straight, right? So we'll see that in these anti-parallel beta pleated sheets here, is that one thing we can look at is we can look and actually see if they're running the opposite direction. And then if we notice those hydrogen bonds to be very straight, because if we look at parallel beta pleated sheets, we kind of see that there's some angles to those hydrogen bonds, right? And these are running the exact same way, right? Carbonyl, behind this green spot is our alpha carbon, nitrogen, and then we have carbon, carbon, nitrogen, carbon. So these are running in the same direction, right? Parallel versus anti-parallel. Thirdly, we have what we consider to be a tertiary structure. So a tertiary structure, right? These are attractive forces between amino acid chains, right? Between amino acid chains. So this secondary structure, this amino acid chain here, this secondary structure is interacting with this beta pleated sheet. And this beta pleated sheet's interacting with this beta pleated sheet. And this alpha helix is interacting with this alpha helix. So we have all of these secondary structures interacting with each other, right? The secondary structures are interacting with each other, and it gives us some functional type of protein now, okay? And we consider those to be secondary or tertiary structures. And lastly, we have what's called our quaternary structure. Now, this tertiary structure And this other red one over here is a tertiary structure. And this blue one is a tertiary structure. And the blue one is a tertiary structure. Here, this was the interaction between two secondary structures to make this. And our tertiary structure is an interaction, quaternary structure is an interaction of these tertiary structures interacting with each other, right? So this tertiary structure is interacting with these, and then we get this one. This one in particular here is hemoglobin, right? Hemoglobin's a tetramer. Now, quaternary structures don't mean that I need 4 tertiary structures. Okay? This one just happens to have 4 tertiary structures interacting with it. Okay, we could have other ones that just have 3. We could have other ones that have just 2 tertiary structures interacting with each other. They would still be considered to be quaternary. So just kind of an overview here, where we see our primary structures is just a sequence of amino acids. Our secondary structures are these alpha helices, beta pleated sheets. Tertiary structures, we get folds here, and these are interactions between secondary structures, and then we have here. So this quaternary structure only has 3 tertiary structures interacting with it. Okay? But we have all of those structures there that we need to identify. Then we have this topic here of what we call denaturation. So denaturation is, yes?
So with those tertiary structures, those certain particular structures, will they be on its own?
It's possible. It's possible.
But
So we,
will they like coiled up like that?
if you were provided an image, could you identify an alpha helix? Could you identify a beta pleated sheet? Maybe which type of beta pleated sheet? Could you recognize that this is a tertiary structure versus something that's a secondary structure? Could you recognize that this is a quaternary structure and not a tertiary structure, right? So just by visualization, if there was an image, could you recognize what type of structure that is? Primary, secondary, tertiary, quaternary structure. Does that help? Okay. So lastly, how we break down these proteins, right? One way we break them down is we call denaturation, right? Thermal denaturation, right? How we break down, what's the cause, what happens when we break down these, because this is different than, I spoke earlier about separating amino acids through hydrolysis. Dehydration to put the protein together, the peptide together, and then we can reverse that reaction and break down that protein called hydrolysis. This is more kind of an unraveling. Okay? Is that if we use heat, we can denature these proteins. Right? Is that here, this is our structure here with alpha helixes, beta pleated sheets, and then we break these down, and then we just get these long ribbon-looking structures. But denaturation is thermal denaturation is not reversible. Once you cook an egg, once you cook a steak, you're not going to uncook that steak or egg somehow, right? Once it's cooked, it's cooked. All of those proteins have been denatured. What's holding this protein together is those hydrogen bonds. The hydrogen bonds keep that helical shape. These hydrogen bonds are what keep this bonded to this, bonded to this, bonded to this. It's what keeps this bonded to this. Without these hydrogen bonds, and they break, they just unravel. And that's what thermal denaturation is. So when we cook something and we break down these proteins, we're not breaking them apart into amino acids, right? But we are breaking all of these hydrogen bonds, and then they begin to unravel. And that's kind of what this image represents here. So this is a denatured protein, right? You ever see those gymnasts that run around with those ribbons, right? Loose, like there's no structure to them at all. So this is a protein that no longer contains hydrogen bonds. But all of the proteins, all of the peptide bonds, the amino acid that's next to this amino acid, that's next to this amino acid, that's next to this one, and all those peptide bonds are still intact. Okay? It's not until we ingest that denatured protein, right? When we cook something, we denature it, we break apart all those hydrogen bonds. Once we consume it, that's when our bodies break that down. So our bodies will take, we take that whole unraveled protein, that denatured protein, and then we start to break them down into individual amino acids that our bodies need, right? Is that goes through hydrolysis. So hydrolysis, we're adding water, right? And we break those down and we get these smaller amino acids. Right, so that's just kind of what we end up doing with those amino acids, or those proteins, as we ingest them, and they undergo hydrolysis. All right, so that'll cover Unit 11. We have one more unit left.